everyone. Welcome to today's webinar on mapping the cellular architecture of the mammalian basal ganglia. Today we have a number of speakers. We'll be using the Q&A tab to go through our questions. And so if you are an attendee, please use that Q&A function to submit any questions, and our speakers will do their best to answer questions throughout the duration of the webinar. And in addition, we'll also have 15 minutes for Q&A at the very end for discussion. If you would like to see closed captioning during the webinar, please click on the Show Captions button down on your screen on Zoom. And this webinar will be recorded, so it'll be going online onto the Allen Institute YouTube channel when we are finished. All right, we can now get started with our first presenter. Our first presenter today is Trygve from the Allen Institute. And Trygve maps the diversity of cell types in the human brain and compares them to other species to understand how our brains have evolved. By pairing large-scale single-cell genomics with genetic tools for precise circuit access, his work lays the groundwork for new approaches to study and treat brain disorders. His talk today is titled Basal Ganglia Cell Type Atlas for Discovery and Tools.
Thanks, Rachel. Can everyone see the slides? Okay.
Yes.
Okay, thank you. And thanks everyone for joining. My name's Trygve, assistant investigator here at the Allen Institute, and been working for quite some time on building out maps of the human brain and non-human primates and also other mammalian species. Today we're focused on basal ganglia, and for good reason. There are, for example, human diseases that affect basal ganglia circuits. So Parkinson's disease, as many may already be familiar, affects these particular population of neurons in the midbrain, these dopaminergic long-range projecting neurons. What we'd really like to know is more about the dysfunction in these circuits, perhaps mechanisms of resilience that could be employed as a future treatment modality. And one path to that is really being able to catalog what is the diversity of neurons in these basal ganglia circuits and how can we target them to study them and again for future treatments. And so, I'll just briefly touch on this and then the rest of the presenters will go into much more detail. But just to say, we're already making some inroads into some of these areas. So this basal ganglia work is part of a larger effort to characterize the nervous system, including peripheral nervous system, a growing effort in that direction, again, across human and non-human primate, and of course, mouse. And this is not only in the sort of neurotypical adult brain, but also understanding early development through postnatal development, aging, and in various disease conditions. And we use a suite of high throughput tools to study the diversity of cells and their properties in the nervous system. And the primary one I'll talk about today is single cell multiomics, single nucleus multiomics, very high throughput method for studying millions of cells. And of course, important to know, because those are dissociated cells, where they came from. And so there's spatial transcriptomic methods that you'll hear more about. And then their anatomical, their morphological and physiological properties. So in particular, in the basal ganglia, we looked at, there was existing data in the mouse. We also profiled human, macaque, and marmoset. Here I'm showing some of the sampling from human and macaque. And these were dissected from some of the core regions of the basal ganglia circuit, shown on the right. So these include, of course, the striatum made of the caudate and the putamen, but also other structures that are linked in a loop, actually, with cortex. And interestingly, there's this really important spatial topography to these circuits, where some regions are more involved in, for example, sensory motor processing and control, whereas more ventral areas are involved in sort of limbic emotional reward processing. And so using these data, we were able to integrate all of the single nucleus RNA-seq data based on shared co-expression profiles across the 3 primate species. You can see that in the lower right of this plot where they all sort of overlap quite well. And using that integrated space, we were able to label these different islands that are shown in this 2D embedding for this couple of million cells. And label these islands based on a lot of many decades of work that has gone towards describing these populations. So, for example, you can see this large D1 island. These are DRD1, dopamine receptor type 1 receptor expressing medium spiny neurons. And then there are D2 MSNs, and then many interneuron populations that are quite rare in the striatum, but very distinct from each other, and then many non-neuronal cell types. And so that's shown in a taxonomic format here. And there were on the order of about 60 different groups that we were able to identify across the 3 species. So, an important component of that, of course, and what actually informed a lot of that taxonomic nomenclature is the spatial transcriptomics, which again, you'll hear much more about. But just to give you a flavor of it here, showing the different types of medium spiny neurons in the macaque, and showing that they have quite distinct spatial localizations. And so we can do this using the transcriptomic identities from the dissociated cells, and then based on a gene panel, map these cells to their to the in situ location. And of course, we can also record the electrical morphological properties. You'll hear more from Ping about this. And we see quite some diversity there as well. And that really will help be the link between, what are these cell types in isolation, and eventually understanding, their function in the circuit. So what can we do with actually just the molecular side of this taxonomy? Well, one approach is to try to understand maybe genetic contributors to human brain disease, or human brain phenotypes. And this is an approach that's been taken in the past to implicate medium spiny neurons, for example, in affective disorders, psychiatric disorders. But here we have a really high resolution atlas where we have many different kinds of medium spiny neurons. And what we find is using an approach that looks for disease-associated genetic variants, these SNPs, from very large GWAS studies of many hundreds of thousands or tens of thousands of individuals, and looks for genes that are near those SNPs and sees if those are markers of these different cell types. And what we find is that these SNPs are not enriched just near markers of MSNs, but actually near the ventral MSNs shown in the middle. And as sort of more of a positive control, Alzheimer's disease genetic liability is really shows up in the non-neuronal cells. And so this was interesting because of the reason I mentioned earlier, that these ventral types are really involved in reward processing and other features of some of these disorders that one can imagine in depression and bipolar, for example. Another use for these atlases is to try to target cell types, and we can use this by identifying these candidate regulatory elements or enhancers, some of which are enhancers, to which very specific transcription factors bind. And those TFs have distinct expression patterns in different cell types in the basal ganglia. And we can find very distinct peaks where the rows are the different groups, and the cells are, and the columns are the different the rows are the different cell types, the columns are the different peaks. And using this approach, we can put this into an AAV and actually label very specific cell types in the brain. And finally, I just want to say, building these atlases has been a large effort internally and with close collaborators, but we really want to open up these annotation and atlases to the whole community. And we're leveraging tools such as the cell annotation platform from the Human Cell Atlas to help enable some of this annotation. So you can go to this URL at the bottom and take a look at this taxonomy we've posted. So thanks to the whole team that has worked on this, involved many efforts of many people, and actually very institute-wide efforts to really bring this all together, and also our many collaborators outside the institute. And thanks to you all. Happy to take questions now or later.
All right, thank you, Trigva. And as a reminder, for questions, please submit them in that Q&A box, and we'll be monitoring the Q&A chat during the duration of the webinar and also at the end. Our next speaker is David Van Essen, who is an alumni endowed professor from Washington University in St. Louis. And David has carried out pioneering studies of the structure, function, connectivity, development, and evolution of the cerebral cortex in humans and non-human primates. This includes widely cited maps of cortical organization and connectivity in the macaque monkey, plus a multimodal surface-based human cortical parcellation. He co-led the Human Connectome Project, a large-scale effort that acquired, analyzed, and freely shared high-quality neuroimaging data from 1,200 healthy adults. The Van Essen Lab has also been heavily involved in the BICAN project since its inception. And today he will be sharing his talk, Navigating the Basal Ganglia Across Primate Species Using Harmonized Ontology in Advanced Visualization Software.
Thank you. Can you see my slides?
Yep, everything looks great.
Great. So I'm pleased to have the chance to talk to you about coordinate frameworks and the common ontology for primate basal ganglia. I'd like to start by expressing my sincere thanks to the many talented people in HMBA who've contributed to this, Washu, Burke, Matt, and Tim, and others at the Allen Institute, Song Lin, Ashwin, Brian, and Mike, and others, Rick and Kobe, to Kuya Hayashi and collaborators, and at INSERM, Henry Kennedy and colleagues. So through these efforts and what you heard about from Trigrid and more to come, HMBA in broad strokes is providing common coordinate frameworks for three intensively studied species, human, macaque, and marmoset. From a structural template perspective, we have MRI-based atlases that provide an anatomical framework and a spatial coordinate system. I'll introduce a novel and powerful approach we are enthusiastic about, what we call the harmonized ontology for mammalian brain atlas, or HMBA approach. You've heard some, and we'll hear much more about the vast amounts of multimodal data coming in through this HMBA endeavor. It includes multiomic and spatial transcriptomics, structural and functional MRI, and postmortem histochemistry and connectivity data. There are also powerful tools for visualization, navigation, and analysis. I'll introduce the Connectome Workbench platform our lab is concentrated on, but you'll hear from others about NeuroGlancer, Cytoexplore Viewer, and other complementary powerful methods. I'll just have time to illustrate a few vignettes involving the basal ganglia. So the templates, as I said, are MRI-based population average volumes. The marmoset and rhesus templates were generated by Takuya Hayashi's lab in Riken. The human template comes from more than 1,000 subjects studied in the HCP project a decade ago. In all three cases, we have a volume average, group average template that has both T1 weighted and T2 weighted images. This is the best modality for subcortical structures. For cerebral cortex, the representation using surface-based templates has many advantages, but I won't have time to discuss that further. So moving on to brain parcels, these are fundamental units of functional organization, be they cortical areas or subcortical nuclei. We'll focus on the basal ganglia first in humans. So here is a parcellation, the Ding Human Brain Atlas generated by Songlin Ding that identified 17 parcels in the human. And working with Ashwin, Songlin and Ashwin developed this harmonized ontology of mammalian brain anatomy that has consistent terminology across species for corresponding parcels. And each species has a set of nested hierarchical levels that are shown in Connectome Workbench in this display on the right, where you can see between the caudate nucleus at the one end of the ellipse and the brain on the other, there are 12 distinct levels of the hierarchy. The utility of this for transcriptomic analyses is shown here. Brian Long and many others at the Allen Institute and others have analyzed the spatial transcriptomic data in a human postmortem brain and identified a first principle component, dominant spatial gradient that's shown in the right here. And then Matt Glasser and Bert Rosen have analyzed the HCP functional connectivity data and revealed a very similar spatial gradient in that modality. Having these two data sets accurately registered to our human MRI template allows us to overlay the HMBA atlas parcellation and see that the spatial gradients are concentrated in the head of the caudate and in the rostral putamen. Using this kind of approach, we can use the DHB atlas in humans to identify not only what the structures are, but where they're locally located. So in this case, if we take the head of the caudate, you can see the crosshairs are centered on the caudate head in each of the three brain slices. But if we're interested in any of the other basal ganglia nuclei, we can easily navigate to whichever one we're interested in. So, for example, the subthalamic nucleus is clinically very relevant, as most of you know. And if we select in our Connecting Workbench visualization the subthalamic nucleus and select a pop-up window, we can jump immediately from the caudate to the subthalamic nucleus. And if we go back and forth, you can see how we can localize in all three brain slices exactly where that parcel is, and we can navigate in that way within species, but we can also use it to compare across species. And for the macaque, we actually have two published parcellations, the SARM by the NIH group and the D99 by a different NIH group that shows these parcellations being very similar, but by no means identical in terms of the original terminology, as well as the spatial location and extent of some of the parcels. And we also have the marmoset parcellation developed by the Salim et al. So using these parcellations across species, Songlin and Ashwin and Burke Rosen implemented a hierarchical representation that for the SARM atlas, you can see where the crosshairs center on the subthalamic nucleus in that example, and in the D99, you can see a successful mapping to that, and in the SAM atlas, corresponding localization. And that can be done for any structure of interest. And this ability to navigate within and across species is going to be, we think, very helpful for analyses of many types of transcriptomic data and other that come along the pipeline. So the take-home lessons basically are that the HMBA and a number of other BICAN consortia are providing vast amounts of high-quality transcriptomic and other modalities of data. These are being accurately mapped to the CCS in ways that I've illustrated. And these resources are intended to benefit you, the neuroscience community, in various aspects of your own research, especially if you use it in combination with new tools for data visualization, navigation, and analysis. And we think that this harmonized ontology, for mammalian brain atlases, the HMBA approach will be very helpful as a part of the resources available. And we certainly welcome feedback from the neuroscience community as to how these tools actually are helpful and how they might be improved in the future. So thank you for your attention.
All right, thank you for your presentation, David. Again, please submit any questions that you have in the Q&A box. And we're moving on to our next presentation by Yan Yan. All right. Are you able to unshare your screen, David?
Yes.
Perfect. Thank you. All right, so while Yan Yan's setting up, I'll introduce her. She is a scientist at the Allen Institute working on single-cell genomics analysis of human and non-human primate brains. And prior to joining the Institute in 2024, she received her PhD at Tsinghua University, where she studied the development of the human brain. And today her title is Cross-Species Consensus Taxonomy of the Primate Basal Ganglia.
Yeah, thanks for introduction. Can you see my slide?
Yep, we can see it. And we can see your pointer as well.
Cool. Yeah, good morning, everyone. I'm Yuan Yuan from Dr. Trigger Bacon's team, and it's a great pleasure to have the chance to present our team's work. So today I'll be presenting the cross-species consensus taxonomy of the primate basal ganglia. Oh. Okay, so before really diving into the taxonomy itself, I would also bring a bit of background, almost all the work, basal ganglia work representing today, actually a part of HMBA, the human and mammalian brain atlas, and the goal actually is to integrate all the modalities, single nucleic RNA, epigenomics, spatial data, patch-seq data, AAV tools, to really try to build a common coordinate framework and to enable us to compare cell types across species and to really link the molecular features with anatomy and function. So within that, the single nuclear transcriptomics consensus taxonomy that our team mainly rely on serves as the backbone. So to build the taxonomy, we sampled nearly 2 million of cells from these core basal ganglia regions across human, macaque, and mammal set, and we conduct 10x multiomics sequencing on them. So here is our workflow to show how we build the consensus taxonomy. We started from macaque, and you can see there are three big steps. We're getting clusters, annotating clusters, then grouping clusters to draft the taxonomy. So to identify each clusters, we began with basic quality control, such as the gene numbers, UMI numbers, and then we do bench correction via SCVI, and we do clustering by HiCat. We also summarize cluster-centric quality control again. So to annotate our clusters, we mainly based on the differential expression genes and also literature markers. As well as the label transfer. That means, yes, we also mapped our clusters data to the existing taxonomies, just providing more additional guidance and context to help refine it. And based on all this information, we merged our clusters into group, which were then merged into subclass and then class and then neighborhood, ultimately forming a draft taxonomy. And this draft taxonomy, our internal teams take it as a reference, like spatial team patch-seq data. They mapped their cells into the taxonomy to help annotate their data, but also meanwhile, their valuable feedback definitely help us to refine the draft taxonomy. And for other species like human mammal set, we basically follow the similar strategy. The main difference lies in step 3, where we no longer do the iterative groupings again to create an entirely new hierarchy. Instead, we mainly primarily relied on mapping against the macaque reference to get the hierarchy. And this way also make it more practical to build the consensus taxonomy. And for the consensus one, species alignment, meta-neighbors, key-ins, and spatial pattern validation, and our others also help. These evidences also help to polish our consensus taxonomy. So here's our core team. Okay, on the left, this is our latest taxonomy. As we said, we have 4 neighborhoods, 12 classes, 36 subclasses, and 61 consensus groups in total. And we summarize the cell number proportion, region distribution per group. And on the right, as Trigger already caught, actually, we truly value the community input. So please feel free to provide any feedback, comments, questions here to help us further refine the taxonomy. Thanks. Yeah. And with the cross-species consensus taxonomy in place, we can start ask a lot of interesting questions. For example, which genes are species conserved and which genes are divergent, or which genes are type specific, which are not. So on the right, we further take a look at the full gene level landscape by plotting the species expression conservation or log values versus cell type specificity tau values, revealing 4 different regulatory regimes. And we also observe some TF transcription factor markers enriched in this conserved gene set. And last, I want to highlight a novel media spanning neuron group emerged in our taxonomy. So we identified the DRD1, D2, and hybrid MSN 3 subclass. But within the D2 MSN, we identified a novel group. We call it dorsal striatum D2 stromate hybrid MSN, since it simultaneously expressed DRD2 and some strosome and classic matrix markers, but they are definitely not doublets. They also has very distinct, uniquely highly expression markers, such as SMYD2, very good pattern conserved across species. And we also identified 2 homologous supertypes in mouse. And the spatial data for the show, actually within this novel group, they have some distinct spatial pattern. For example, this supertype, the green dots, only enriched in ventral, and a similar pattern were also observed in our primate spatial data, here colored by red dots. And this ventral biased subgroup also shows some conserved high expression markers such as HTR7. So due to time, I'll stop here, and I want to thank all my collaborators and apologize I can't call out everyone, but this is definitely a teamwork. Thanks.
All right. Thank you, Yan. We'll be moving on to our next presentation, which will be by Maddie and Megan. Maddie and Megan are both from the Allen Institute, and Maddie has been working at the Allen Institute on spatial transcriptomic analysis of the human and non-human primate brains. And prior to joining the Institute in 2023, she received her PhD from the University of Washington, where she studied cell shape of zebrafish sensory organs. And for Megan, Megan is also a scientist at the Allen Institute, also working on the analysis of spatial transcriptomic data in human and non-human primate brains. And prior to joining the Institute in 2023, Megan earned her PhD in biophysics from the University of California, Berkeley, where she studied the molecular mechanisms of transcriptional regulation during embryonic development. They'll be presenting a talk today together titled A Cross-Species Spatial Transcriptomic Atlas of the Human and Non-Human Primate Basal Ganglia.
Is the presentation showing up appropriately?
Yes, it looks good.
Okay, great. Thank you for that introduction. I'm Maddie, and I'm going to get us started with this presentation. So why do we do spatial transcriptomics? Well, one analogy is that it's kind of like a jigsaw puzzle. So when you first get the puzzle, all of the individual pieces are mixed together. And you can sort the pieces by features like their color or pattern to start to understand how they're related to each other. But you really need to assemble the puzzle and put the pieces in their proper spatial context to get the full picture. Similarly, single-cell RNA sequencing technologies allow us to group cells based on their gene expression and create cell-type taxonomies to understand how cells are related to each other. But this doesn't give us the full picture, and we really need spatial transcriptomics to see how the cells are actually physically related in space, which reveals patterns like the fact that the striosome MSNs in red form distinct patches from the matrix MSNs in blue in the striatum. Once we have the cells in their proper spatial context, we can start to link them to other data modalities, such as structural MRI, as David was talking about earlier. And so using spatial transcriptomics to bridge the gap between the single-cell taxonomies and other data modalities like MRI could eventually help us to understand the relationship between cell type, structure, and function in the brain. So that's broadly why we do spatial transcriptomics, and now I'm going to talk about the goals for this specific project. We want to understand the spatial organization of basal ganglia cell types in 3 primate species, the marmoset, macaque, and human, and identify themes that are conserved and divergent between species. And also, we want to provide a resource for viewers like you to explore your genes, cell types, and regions of interest in the BG. Because imaging-based spatial transcriptomic platforms have fixed-size gene panels, we could only profile 300 genes per species. To enable cross-species comparisons, we designed partially overlapping gene panels. We start by manually selecting marker genes that define broad key cell type populations, like non-neuronal versus neuronal marker genes. Then we use computational tools to fill out the rest of the gene panel, selecting genes that are differentially expressed between the finest resolution cell types, called clusters. To confirm that our gene panel can identify cell types, we perform a self-mapping assessment, where we use just the 300 gene spatial panel to map the reference RNA-seq data back to itself. We use a confusion matrix where more types on the diagonal is better to check that the labels when using just the 300 genes matches the full gene set reasonably well. Originally, when we get the data, it comes off as raw transcript locations. And to assign these to cells, we have to do cell segmentation and quality control. For the segmentation, we use stains like nuclear DAPI in all transcript images to identify the likely boundaries of each cell. We then filter out the low-quality cells shown here in red based on metrics like total transcript counts. While the marmoset brain is small enough to capture a full hemisphere in one experiment, for macaque and human, we have to cut them into smaller blocks to fit in the available imaging window. So this is where Mike Huang basically does his own jigsaw puzzle and stitches the blocks together and registers them to place the sections in their proper anatomical orientation. After the segmentation and quality control, we map each spatial cell to its best matched cluster in the basal ganglia consensus taxonomy using Map My Cells. This allows us to analyze the location of cell types and provide feedback to refine the annotations in the taxonomy. For the analysis, we focus on the group level, which is the finest resolution level of the taxonomy that is conserved across all 3 species. And now I'm going to hand things off to Megan, who's going to show more about the data and results.
So now that we have our spatial atlas all put together and QC'd, this allows us to visualize the spatial locations of the exact same cell types from the consensus taxonomy in all 3 primate species. In these montages, we're showing the full rostral caudal extent of our data collection in each species, with the rostral displayed at upper left and the caudal end displayed at the lower right in each species. Despite differences in scale and sectioning resolution, we've collected more than 1 million basal ganglia cells per species. And now with this comprehensive data set in hand, we've leveraged our spatial transcriptomics data to answer outstanding questions about conserved spatial organization of the basal ganglia nuclei across these 3 primate species. As just one example, and Maddie kind of alluded to this before, we find remarkably similar organization of cell types in the striatum across marmoset, macaque, and human, with specialized medium spiny neurons localized to the ventral striatum, shown here in green, and further specialization of the dorsal striatum into striosome MSNs that float as little patches in the matrix MSNs, shown here in blue. One of the other things we can do with our spatial atlas is we can now use the cell type labels on these spatial cells to create polygon masks that outline the boundaries of the discrete compartments of the striatum that are known to exist. So here we reveal the striosome, shown here as red patches that form the patches inside the matrix, We also see the islands of Calleja in orange and the NUDAP interface islands in pink. In addition to these cell types and compartments, we find a dorsal ventral gradient that is superimposed onto this discrete structure of the striatum. This spatial gradient is a complex combination of multiple genes, and we can identify these gene sets using principal component analysis, with the corresponding dorsal ventral spatial component shown here from red in the dorsal striatum to blue down in the ventral striatum. When we put these different layers of organization together, we can now see that the striatum is simultaneously defined by discrete compartments and continuous gene expression gradients, and that these spatial patterns are conserved across all 3 primate species that we sampled. In this short webinar, we were only able to show you the striatum, but across all the different basal ganglia nuclei that we sampled here, we find that the molecular organization is highly conserved across primates and often even shared with rodents. This conservation is maintained whether the organization of the basal ganglia nucleus is defined by discrete compartments or continuous gene expression, or in the case of the striatum, both. And the analysis tools we've developed for this first phase of HMBA, which was just focused on the basal ganglia, will be transferable to the entire brain, an effort which is currently ongoing. Now, if you're just beginning to work with spatial transcriptomics data, you might be wondering where to start. Here are some tips that we've learned from our project that we hope can help you with yours. First off, spatial transcriptomics is still an emerging technology, and the field is still working to develop best practices for analysis. So pick something for now, run with it, and then iterate later once you've learned more about your own data. Start with established supported tools. We've included a number of our favorites for various purposes below, and then go and build your own tools only if critical functionality is missing from these existing ones. Look at your data. You have the benefit of spatial context. Trygve and Yuan alluded to this, but it's really helpful to use the spatial data to help you understand where your cell types are localized and what their identities might be. This really does give you a good feel for what looks right and wrong in your data. And if you're not sure what you're looking at, it might just be in a histology paper from the 90s or one of the many ISH atlases that the Allen Institute and other institutions have put out and published for your use. Of course, this cross-species transspatial transcriptomic atlas was a huge multi-year project that was really only possible thanks to the contributions of many, many people at the Allen Institute, as well as all of our BICAN HMBA consortium collaborators across many universities and institutes. And we'd like to acknowledge their hard work now. And thank you all for your attention to this webinar. I'll turn it back over to Rachel.
Great. Thank you, Megan and Maddie. I especially appreciate all those helpful links you had on that last slide. And if anyone missed those, don't worry. Again, this webinar will be going up on our YouTube channel later today, so you can have access to those links again. All right, our next presenter is Ping, and she is also from the Allen Institute. She is on the human cell types team, where she performs quantitative analysis on patch-seq data, which provides multimodal single-cell RNA sequencing, electrophysiological, and morphological characterization of neuronal cells. Previously, she completed her PhD in the joint Harvard-MIT Health Sciences Technology program. She has studied neural processing of auditory stimuli, maturation of mechanotransduction, and electrophysiological properties in inner ear sensory cells and ion channels of inner ear sensory cells. And today she'll be presenting morphoelectric diversity of primate striatal neurons revealed by patch-seq.
Hi, can you hear me?
Yep, we can hear you. And we can see your pointer as well on the slides.
Okay, great, thank you. Okay, so we've built this wonderful atlas, but how can we put functional phenotypes on each of these types of cells that we've identified? One way is through a method called patch-seq, where we first use patch clamp to form electrical continuity with the inside of the cell, and we deliver electrical stimulation and record the responses. There's also a biocytin dye inside the pipette, which allows us to get recovery and morphological reconstructions. And then at the end of the experiment, we aspirate the nucleus and some of the cytoplasmic content, and then we send that out for transcriptomic sequencing. And this allows us to annotate the functional properties of neurons anchored to their transcriptomic type. I also want to point out that while much is known about the in vitro properties of striatal neurons in rodents, relatively little is known in primates. And since the basal ganglia is not a structure for which there is availability of human surgical resections, we had to rely on non-human primate tissue distribution programs, which provide tissue from macaque monkeys that are slated for necropsy. Okay, so what were we able to learn from this data? First, we can get a high-resolution look at the principal neurons of the striatum, the medium spiny neurons, which have traditionally been classified as either as D1 or D2 receptor expressing. We can see systematic variation, for instance, between these bushy MSNs of the dorsal striatum versus the sparse MSNs of the ventral striatum. There are also a number of non-canonical MSN types, the D1, D2 hybrids, and the D1 nudaps that share this third island over here. And then also this group of D2 striomat hybrids, or D2 hybrids for short, which is a distinct set of MSNs that co-expresses striosome and matrix markers. And you can kind of see from the UMAP already that these may not really be truly hybrids of other types, but their own distinct groups. And indeed, the properties of non-canonical MSNs differ from those of other MSNs. For instance, they have lower spiking thresholds, more low-frequency filtering, and a more pronounced slow component of the after hyperpolarization following each action potential. We had very few D1 UDAP samples, but they also appear to have eccentric properties. Here I've made a UMAP embedding. Instead of considering one electrophysiological feature at a time, this is a two-dimensional representation of the electrophysiological variability across features. It's kind of hard to see, so I've created kernel density estimate contours showing where D1 matrix, D2 matrix, D2 striomat hybrids, and the D2 hybrids reside in the space, and you can see that the hybrid types have properties that are rather distinct. Okay. So another really interesting group of cells. Oops, I missed one. Okay, so on this same UMAP, we can highlight everything in the D1 MSN subclass, which includes matrix, striosome, and ventral types, as well as the D2 MSN subclass. I will not go into details, but basically both in terms of electrophysiological and morphological differences between D1 and D2 MSNs, we don't see the same differences that were reported in rodent, or in some cases, it's in the same direction described, but very subtle. However, there are clearly differences between D1 and D2 MSNs, as you can see from MAP. And if we perform a two-way ANOVA, taking into account both D1 versus D2, matrix versus striosome, we also see a number of significant differences. And although there are consistent differences, you can see that the ranges are overlapping. So another fascinating cell type that we looked at was the granule cells of the islands of Calleja, which have been implicated in reward, addiction, and depression. Relatively little is known about these cells, even in rodents. They're incredibly tiny and hard to patch. Our data show that although they may be transcriptomically related to MSNs, their properties are quite distinct, with a very high input resistance, low urea base, and low capacitance, consistent with a very small cell, and consistent with reports in rodent granule cells. However, to our surprise, the majority of our primate granule cells exhibited bursting. The prevalence of bursting in granule cells may turn out to be a species difference. Okay, because the majority of our samples are pinned in 3D space via photo documentation within an anatomical reference, we can examine spatial variation in properties within a subclass. What we see is reminiscent of the gradients observed in spatial transcriptomics, going towards and away from the nucleus accumbens pole. Intriguingly, several of these are similar for MSNs and fast-spiking PTHLH interneurons. We don't have enough cholinergic interneurons to do this kind of analysis. When we just compare dorsal versus ventral cholinergic types, we see similar morphological and electrophysiological shifts, which can be summed up as more sparse morphologies and slower action potential kinetics ventrally. A recently described group of interneurons expressing the TAC3 gene appears to be highly divergent between mouse and primate, although most closely related to the TH-expressing neurons in rodents. They have spike width intermediate between the fast-spiking interneurons, marked by the gene PTHLH, and the regular spiking interneurons marked by SST. You see that although they can achieve some relatively rapid instantaneous firing frequencies, they're not able to sustain that firing. And their firing rate averaged over the entire step does not keep pace with the fast spiking PTH-LH neurons. However, they have relatively similar basket-like morphology. Lastly, we collaborated with a team that was performing patch-seq using the same protocols in mouse basal ganglia, and we're able to make some fine-grained cross-species comparisons. In short, there are numerous differences between the species. This list here is truncated. We also had some data from Cymeri, which is squirrel monkey, that could be used as a New World primate comparison. These differences include the elaboration of primate cholinergic interneuron morphologies compared to rodents, species differences in both fast spiking PTH-LH interneurons and TAC3-TH interneurons, the slower component of the HP that distinguishes D1D2 hybrids from other MSNs in macaque, but not mouse. So if you focus on the green, you can see that this is differential for D1D2 hybrid in macaque, but not particularly in mouse, and more low frequency filtering in primates. Oh, there we go. Okay. So to summarize, medium spiny neurons have underappreciated and multiplex phenotypic diversity mirroring transcriptomic variation, but with substantial overlap between groups. We should move beyond a binary model of medium spiny neurons. D1, D2 hybrids, and D2 hybrids have unique properties that warrant further investigation. Alonso-Callejo granule cells frequently have a bursting response in primates. A spatial gradient can be seen in several morphological and electrophysiological features that may be concordant across multiple subclasses of neurons. TAC3 interneurons have divergent properties in primates and are unable to sustain firing under sustained depolarization. Numerous other differences exist between homologous groups in rodents and primates. Okay. And this is the work of a large team of researchers at the Allen Institute who I've had the privilege of working with. And these data will be made available for exploration in the CytoExplore viewer, which you will hear more about shortly.
All right, perfect. Thank you, Ping. We can now move on to our next presentation, which will be by Bodwin, who is a professor of biomedical engineering in the Department of Radiology at Leiden University Medical Center and at the Pattern Recognition and Bioinformatics Lab at Delft University of Technology, the Netherlands. His team focuses on development of fast algorithms and software for highly interactive visual analytics of integrated single-cell and spatially resolved omics data, and he'll be presenting Manivault Studio and CytoExplore Viewer. For HMBA specifically, his team developed several CytoExplore viewers for cross-species comparison, data integration, and cross-model visualization of the HMBA data.
Thank you very much, Rachel. I'm very happy to present some of our contributions to the HMBA project. And I'm presenting this on behalf of a team that has been working diligently over the past couple of years on developing this software. And here you see the people actually a part of the team. So I'm myself in a medical faculty, but I am a computer scientist, and I'm also affiliated to a computer science faculty in TU Delft. And this has been a collaboration between these two universities across the past couple of years. So our main research between these two universities is at this point like on visual analytics for high-dimensional data, and this was sparked by Laurens van der Maten, who actually developed the TSNE algorithm, and together with him, he was in Delft. We actually developed a research line to actually make this dimensionality reduction techniques fast and scalable, and to deploy them in a way that they become usable for interpreting this high-dimensional heterogeneous data set. So there's many algorithmic and developmental work that has gone into this over the past couple of years. And really what we try to accomplish there is to make it into a visual analytics application. And visual analytics goes a bit more than just data visualization, where visual analytics really kind of allows you to select subsets, to drill into that, to look at differential expression, and to really interact with the data. You can pick up the data and shake it and and really kind of get an idea of what's going on there and to really query that. And to be able to do that, we developed this cytospore application already back in 2016. And that was really for people in immunology looking at their own data. But since 2018, we have been developing Cytospore Viewer, and Cytospore Viewer is really tailored to large-scale curated and archival data sets, such as the one being generated in HMBA. And we were involved in a number of the earlier big data releases of the Allen Institute, developing specific viewers that really would cater to bring out the most of the data and to enable users to work with it in a very interactive way. And that is actually how we entered into HMBA, where we really try to look for a complementarity between what we do and the large-scale web-based enterprise platforms that the Allen Institute is developing. So the ABC Atlas and the Brain Knowledge Platform, they really can cater for enterprise-scale data hosting and handling that simply universities would not be able to do. And to do this in a web-based way and actually use this to really establish reference standards and common coordinate systems. And whereas the visual analytics component from our Cytospore viewers allows an interactive compute, and we really try to focus to combine multiple species and or multiple omics modalities. And that would allow you to, for instance, drill into detail, but also to zoom out again, to do re-embedding and all kinds of things that require a local compute that has to be very fast and very interactive, and differential expression and everything on the fly. And really allowing you to go all the way from the overview down to the single cell. We want to be able to even look in the cupboard in the kitchen to individual cells and to measure them and to interact with those. Now, and here enter the basal ganglia taxonomy that has been presented today. For us, this has been really instrumental and very, very productive, because this taxonomy, if all data is mapped to the identical same taxonomy, it becomes much easier to connect everything to everything. And we really try to concentrate on developing viewers that would go across modalities or go across species. For instance, we developed a viewer where we would actually be able to drill into cross-species differences in the single cell data that we saw today. But we also have a viewer that connects electrophysiology and morphology and T-types as we just saw them. We have a viewer that connects single-cell RNA sequencing data to spatial transcriptomics to ataxy, chromatin opening. And we have a specific viewer for comparing spatial gradients and how do they behave and how are they conserved across the three species. To give you an idea of one such viewer would be, this is a viewer that we made for PET-seq data. You can actually easily interact with it. You can select cells in there. You see the morphologies of the cells. You see the electrophysiology of the cells. And here you can actually see the selected cells, what are the features, the properties that make them unique. And this is all in a very much interactive manner where you can make these selections in a very dynamic way. And you can, for instance, select cells in a morphological UMAP and then actually see which electrophysiological features distinguish these cells that you just selected from the morphology. And so they are similar from morphology, and what are the electrophysiology features that set them apart? As an example of how we try to go across modalities. Here you see an example where we can look up a gene and actually project it with spatial data, and then we can select a region of interest, and we get attack peaks, chromosome locations that exhibit a very similar spatial profile. So we can really do bidirectional searches between spatial gene expression and chromatin opening patterns in the spatial domain. And this all in a highly responsive and interactive manner. We also have, as I mentioned, like a drill-in viewer, and this is always challenging to do something live during a webinar. But this allows you to, for instance, select cells, and then on the fly we can press, we can actually generate TSNE. We can drill into subparts of the mapping TSNE maps, and we can color code cells We can select cells and do differential expression. So now we have two selections and we have differential expression, and you can then click on the genes that are differentially expressed to actually inspect them really live on the fly and compare this across the different species. And yeah, a final tool, as I mentioned, is really something that we came up with together with the spatial team. Is that really it's all about the gradients in the spatial data, especially in the basal ganglia. That we made something that makes it really easy to actually identify genes that show a spatial gradient, and then actually compare across species whether these gradients are actually preserved across different species. Yeah, I put in these live images just to show that this is kind of live interaction as it can take place. You can download this software from viewer.siteexplorer.org. It's yeah, application based, so there we have a Windows and a Mac installer, and yeah, all the data is hosted at the Allen Institute Amazon bucket. And the data handling and downloading is all taken care of for you. You have to download it once, and then you can work with it yourself on your own medium end hardware, let's say. So that concludes my presentation. Thank you very much.
All right, thank you, Bodwin. Our last presenter of the day is actually myself. I'm Rachel Hostetler, and I joined the Allen Institute in 2023 after finishing my PhD in neuroscience at West Virginia University, where I studied the diversity of cortical somatostatin inhibitory interneurons. And here at the Allen Institute, I'm part of the scientific and public outreach of cell type taxonomies team, which means that in my role, I provide training, technical support, and outreach to scientific users of Allen's cell type taxonomy data and tools. And I'll be presenting today a talk titled Exploring the Basal Ganglia Taxonomy through the Allen Brain Cell Atlas. All right, does everything look all right with my screen? Okay. So we actually recently just remodeled our website at the Allen Institute for Brain Science, and I want to highlight that quickly because we've talked about a lot of different resources and data sets, and sometimes it can be quite difficult to find all of these things. So if you go to brain-map.org, that is the main website for the Brain Science Division at Allen Institute, which houses all of our data and all of our tools. And with part of our new website design, we actually have this new search bar here where you can just search in anything you'd like and find data and tools much easier and much quicker. And so if you just type in HMBA, you can actually find a landing page that we have on our website featuring all of these data and tools. And so we have the basal ganglia, which is the first release here of that human and mammalian brain atlas. And then additionally on this landing page, we also have information about the preprints that will be available that are associated with this current paper package. And there's a list of them down here in the bottom left-hand corner. In addition to learning more about the data and about this project and why we're doing it, we also have links to a lot of the tools that you've heard about today. So we learned from David Van Essen about HMBA, or the Harmonized Ontology of Mammalian Brain Anatomy. There are also plenty of links to these data sets and to these tools here. If you'd like to download the data, that's all accessible here on that landing page. We just heard about CytoExplore. We also have links to CytoExplore on this page as well. And we also heard about the Connectome Workbench earlier today for visualizing our common coordinate frameworks and transcriptomic samples. And these are just some of the tools that are linked on this landing page. There's many more tools, but I just want to highlight that this is really a good place, a one-stop shop for all of the tools and technologies and data sets and tutorials and resources related to the project that you've been learning about today, the Basal Ganglia Atlas. And that's all available at brain-map.org/consortia/HMBA. Or you can just go to brain-map.org and just type in that search bar HMBA, and you will find all of these resources. So in addition to having all those resources on that page, we also have links out to the ABC Atlas or the Allen Brain Cell Atlas. And we heard a little bit about this atlas from the previous talk by Bodwin, where it's this enterprise level web-based application tool developed by our data and technology team here at the Allen Institute. And the ABC Atlas is about 2 years old now. It contains multiple data sets from mouse, human, non-human primate, and it now features all of the basal ganglia data, the transcriptomic data that we have talked about today. So unlike Cytosplore, which has patch-seq data and some other modalities, the ABC Atlas really, for now, features single-cell data and spatial transcriptomic data. But here in this video, I'll show you how we can access some of the single-cell data. So this is all from that landing page that I previously mentioned. Here's what the interactive viewer looks like. And what we see here first is that UMAP, which is showing that consensus taxonomy here. And we can see our different species. If we go down and look at genus, we can see our human, macaque, and marmoset coloring. We can look at the different dissection regions of interest here. This is all color-coded now by those different dissection regions. And then with that consensus taxonomy, we can go through the different levels of the taxonomy. So at the very high level, we have 4 different neighborhoods. And then within those neighborhoods, we have our classes, we have our subclasses, we have our groups, and then we have our clusters. And you can actually go through the taxonomy at these different levels, coloring the UMAP and digging deeper and deeper and deeper at these different levels. And as you can see here on the left, all of those classes, subclasses, everything else, they're nested within, their parent function. And by changing the color, we can learn more about where they are present in the UMAP. And if we go down all the way to the group level, remember the group level is where we have that consensus taxonomy conserved through our 3 different species. And we have some panels here to give you more information about the acronyms used, which level of the taxonomy you're at, the parent and the children. And down here at that cluster level, we can really see those species-specific clusters that are nested within those conserved cross-species groups. And you can also just isolate a few of them as well. So right now we're coloring by all those different clusters. And then I click on one of the clusters just to say, okay, let's just focus on this individual one here. And then it'll filter out that cluster on the UMAP. You can zoom in, get more information. You can go back and look at all of the data together. Again, right now, it's still colored by cluster. And we heard earlier that there were some marker genes involved in some of these studies. So you can actually view the different marker genes that were used at different levels of the taxonomy. And so right now here, I'm just filtering it by that specific group in the dorsal striatum, our medium spiny neurons. And I want to view those marker genes. So you can actually view the individual genes in the ABC Atlas as well. I just did that. I clicked add set to genes, and now I can view the genes in the specific genes in the UMAP. Now I'm going to go and clear out my filter so we can see the entire UMAP again. And again, we can look at all those different genes. We can type in any of the 1,600 genes in the data set and just view their expression across the entire UMAP. And in this image here, darker color is higher expression. We also have a new ABC Atlas AI assisted chatbot here, which was just launched a few weeks ago. It's still in beta mode. But you can ask it questions. For instance, you're curious about what data you're looking at. Maybe you don't remember which gene you picked. It can tell you information about the plot that you're specifically looking at. Not only can it tell you information about what you're looking at, it can also create new plots. So you can give it instructions and say, show me this gene, show me this type of plot. I'm interested in this type of data. If you ask a question that it cannot do, like for instance, it cannot do computations on the fly, if you try to ask it that, it will tell you politely, that is not one of my functions. But currently right now, this new AI assistant is really just used for navigating the interface of the Allen Brain Cell Atlas. And a lot of this work was done by Shane Vance, Chris Morrison, Sue Lee, and Pierre de Valentin from the data technology team at the Allen Institute. And so right now I'm just having it show different types of plots colored with different genes, and then also make me a third plot and show me different cell types and groups. And you can have up to 4 plots side by side in the Allen Brain Cell Atlas. And since these are the same plot, you can actually link them together, sync them, and get a closer view diving deep into a specific area of the UMAP that you are interested in. And when it pops up and highlights those different colors in the different plots, you can see that those are the same cells that are in the different UMaps. All right, so that's just a brief example of how you can interact with the single cell data in the Allen Brain Cell Atlas. But as we learned from Maddie and Megan, we have a lot of transcriptomic data, or sorry, a lot of spatial data as well. So here's just a brief video showing how you can interact with some of these spatial data. So the spatial data is laid out a bit differently. Remember, we have our human, our marmoset, and macaque. So those are present here as 3 different windows. Human on the top, macaque bottom left, bottom right marmoset. I'm just going to focus on one of them just to clear some space. So I'm just going to highlight the macaque data. And if we zoom in here on the spatial data, right, as we learned earlier in the previous talk, right, it's sort of a jigsaw puzzle registering all those sections together. And so each of those sections has a specific colored label. But you can also color the different cells by their manual annotations, so their anatomical regions. And also that same taxonomy again, right? It's the neighborhood, class, subclass, all the way down to clusters. That's the same in the single cell data as it is in the spatial data. And so if you go through here and look at all the different groups and subgroups, it's the same names and cell labels that we just saw in that single cell data. And we can zoom in and view now those spatial locations of those particular cell types. And here, since we're looking at the macaque data set, here's where we have those macaque-specific clusters that are shown in our spatial data as well. And here I briefly recreated a figure that was shown earlier by Megan, where you can really see the differences in the stereosome and other cell types and the other different islands. And so this is just another way to view and interact with these data sets. Right now I have both of these plots. I have marmoset on the right, I have macaque on the left. And we can view all these different cell types and actually see where they're conserved spatially across those 2 different species. All right. The atlas that I just showed you, the Allen Brain Cell Atlas, is really for data visualization. So if you would like to download and access the data, we have a number of other ways to do that. You can access the data programmatically here through our ABC Atlas Python library. A lot of this is maintained by Chris Morrison from the Data and Technology team. And we also have some other options for downloading and browsing and visualizing data. We have another tool that was mentioned a bit in the earlier talks called Map My Cells, where you can actually map your data and annotate it with the cell labels that were used in that basal ganglia taxonomy. And as you heard from the previous talks, this was actually used a lot in the development of this project. And this is online for anyone to use. We have a nice user interface here where you can click on Map My Cells. We also have example notebooks. So if you just want to run the code yourself and don't want to use the interface, again, that is one option. But here's what it looks like in the browser. It's very simple, drag and drop. If you use the browser tool, there are some limitations for file size and the type of file that you're uploading. But here we have a number of different taxonomies you can map to. Here's that consensus basal ganglia taxonomy. And these are some really helpful links just explaining more about the tool, how it does the mapping, the input and output files, some example data sets that you would like to use, and how to create specific files using Python or R. And here in this link as well, we have some more information about just the specific taxonomies that are used, how the mapping algorithms were created, and just more detailed information. And here we're jumping down to the basal ganglia consensus taxonomy, again, where you can read more information about how this taxonomy was created. And again, we have more information on our GitHub, many GitHub repos as well. There's a link to that where you can actually just access the code and run it yourself if you would prefer that. All right, so as I mentioned before, all of this is on brain-map.org, where you can just search HMBA, or you can go through consortia/HMBA. And we also have a community forum, so community.brain-map.org. This is a really useful resource where many of us at the Allen Institute are checking this on a daily basis, trying our best to answer questions from users in the community. So if you have an issue with perhaps your mapping algorithms using Map My Cells, something crashes, you're having difficulties with one of these tools, you can please post in our community forum, post your questions, search through other people's questions, and it's just a really useful interactive resource to be able to help users understand and better use these tools and data. And it also helps us at the Allen Institute know which features are useful, which features could be more useful. It's really helpful to get that community feedback. All right, thank you very much for listening to this. And my presentation is actually the last talk. So now we can actually move on to the Q&A section. All right, if all of the panelists would like to come back online or come back on the videos. Let's see first if we have any unanswered questions in the chat. So there is one question that we have here. Is there any work being done on plasticity, both short-term and long-term, being done in the consortium? And any work specifically on neuromodulation, for example, dopaminergic, cholinergic of this plasticity? And this could be a free-for-all answer for anyone to answer regarding BICAN work.
I don't know, Ping, if you want
Um, yeah, my hold on. My understanding is, so by plasticity, I assume you mean synaptic plasticity. I don't think there is work being done right now on synaptic plasticity. Sorry, question just disappeared. And in terms of neuromodulation, I believe there is some work going on, but it's not necessarily focused on basal ganglia.
Yeah, I mean, I would just say that, we're building up these maps to be able to ask these very questions. So, I think as we get toolkits to target these neuromodulatory systems and then, there's the whole community, right, is hopefully going to follow up on all this. But yeah, we're starting from the parts first and then circuits.
Yeah, and that leads a bit into the next question. What are some lessons learned from building an atlas of the basal ganglia that can be extended to the whole brain?
I mean, maybe I could just briefly comment, and others can chime in. I think, just how we laid out the progression from starting from tissue and going to cell types, mapping those cell types in their spatial context and understanding their properties, that required a large amount of work behind the scenes to define standards, data standards, ontologies, mapping to the anatomical framework that David Van Essen described. And I think all of that kind of sets us up to then do this for other brain regions. Yeah, so and there were just many challenges along the way that had to be resolved to make that all work.
I think another interesting thing when I think of the spatial transcriptomics is just the size of everything, right? Going from whole mouse brain where it can fit on a section versus moving up larger in size, you have to make these different divisions of your tissue, right? You have that jigsaw puzzle, and that's only going up the whole brain, right? That's only going to get more complex. Do we have anyone else who would like to comment on that aspect?
Yeah, I think like the thing we have learned is the fewer cuts you can make, the easier life will be stitching it back together. So that is certainly, I think, a shift to the whole brain as you start thinking about the feasibility of that. The less blocking we can do, which is what we call cutting it kind of into those puzzle pieces, less blocking we can do, the better. And that's kind of a shift that'll be happening for the larger brains, for the whole brain effort, is to try to get bigger things on bigger slides so that we don't have to do quite as much stitching back together.
For patch-seq, we're still not a very high throughput method, so I think we would have to focus on particular brain regions.
We have a question specifically regarded to the patch-seq data. The patch-seq data that was presented today, is that up on bioRxiv yet? Someone is asking to see more information about that.
Yeah, so we are in the final stages of trying to write this up. So please be patient, and yeah, the intent is to make it available.
Yeah, and I mentioned during my section that we have that landing page where once everything is up online, all the preprints are up on bioRxiv, that will be linked there. So if you go to that landing page, we will be updating that continuously just to have everything in one place. All right, another question we have is, really cool visualization tool. Thank you for the collective work. I'm new to the Allen Brain Cell Atlas and wondering if I can find full names for the region of interest groups. So I know that for when looking at the mouse data in the ABC Atlas, that all those same acronyms are used from the mouse reference atlas that we have, and same with the human reference atlas as well. Do we have any other thoughts or comments about the marmoset and macaque data, right? These are all the same acronyms. So maybe any other easier ways to understand or links for understanding these different acronyms. I will say that the data and technology team is developing a new feature in the Atlas where it'll have a button that says learn more, and then they'll have that pop-out panel that actually lists the acronyms that currently exists for the cell type names. I demonstrated that a little bit in the demo, but right now that doesn't exist yet for the regions of interest, but that is something that the data team is aware of, and they know that that's a wanted feature in the tool. So hopefully we'll see that in the future.
Yeah, so there is on the GitHub tutorial page, I'll quick drop it as a response there. There is an HTML page that allows you to get all of the HMBA, like unified ontology for all of the regions. And it's interactive, like it'll expand out the different levels of the hierarchy, and it has both the acronyms as well as the full names, and then how they all relate to each other within the hierarchy. And there's a nice little search bar. So if you know either the acronym or a part of the full name, right, that you're looking for, like you don't know what acronym we use for it, you can use that search bar, and it'll kind of filter down the hierarchy and collapse it to anything that kind of relates closely to that search term. And yeah, I believe this one on the GitHub page is the stable version of it, but someone else can correct me if I'm wrong, and there's a more stable link currently available.
And the link to this should be on that landing page that I mentioned, that HMBA landing page that does have that section on HMBA and a couple of different GitHub links as well. But it is good feedback. We would like to actually integrate that in the Atlas itself so that you don't need to have multiple windows open. That is something that we're aiming for in the future. All right, let's look at some other questions that we have. This is a question specifically that could go to David. Do the parcels identified in the harmonized ontology of HMBA always represent structures that are conserved during primate evolution?
So that's a good question. And if you take any of the fine-grained parcels like the globus pallidus exterior or the substantia nigra, which has different component parts, the working hypothesis and the objective of the HMBA concept is to identify anatomical structures that correspond not only in terms of their functional organization, but in having a common evolutionary origin. However, that's really a hard problem to solve definitively. So it really depends on expert neuroanatomists and neuroscientists to look at the data and say, is this structure in a macaque parcellation actually the same entity based on a common set of features, not only their transcriptomic profile, but their architecture, their connectivity, and if you have access to that information, to their functional characteristics. So it's highly likely that some of the assignments made in the current HMBA will turn out on closer review to need refinement. And so this should be viewed as a work in progress and essentially a set of hypotheses about what are correspondences that need to be scrutinized uh, by the generators, but also by the community at large to chime in and say, this particular subdivision of the Susantia nigra may need reevaluation, reclassification when additional experimental data becomes available. So the point is to emphasize this is a dynamic process, not a static state of known truth. This is a set of working hypotheses that the community needs to understand and engage in evaluation and refinement of.
Great, thank you. Another related question is, what are some properties of cell types that will be valuable to add to the current atlas that aren't there right now?
Connectivity. Local and long range. We'll get there.
So did you say connectivity?
Yes.
Yeah, I fully agree. Critical to understanding, but hard to get. But it's part of the work in progress. Henry Kennedy's lab, for example, is providing valuable information about connectivity revealed by retrograde tracers. But that's a work in progress. But it will help inform these issues for sure.
Now we have a few questions specifically related to the Cytoexplorer viewer. Is there any plans in the future for making perhaps a web-based version of the viewer? And is it possible to integrate other HMBA data sets into Cytoexplorer?
So basically, the answer for the web-based platform is actually no. So we actually deliberately chose for, let's say, application-built Site Explorer because it really leverages the local compute and the interfacing across different data sets. And also, yeah, we try to be as complementary as possible to the ABC Atlas and Brain Knowledge platform. Having said that, it is possible to actually integrate new data sets in there, especially if they're mapped to the same taxonomy. Then, as I showed before, then it becomes much easier to actually connect things and kind of mine across different modalities, like, for instance, the spatial mapping of attack peaks, and then using that as kind of a search template for spatial gene expression and kind of correlating the two between directly different modalities. And that could even serve to link on one hand the GWAS world, the gene coordinate world, to the RNA-seq gene expression world in that sense. So if data sets are mapped to this coordinate system, it becomes certainly feasible to integrate it in the Site Explorer viewer.
Yeah, and those are some great points that are actually questions that are and features that are asked about ABC Atlas. People ask, can I do computations with it? Can I integrate my own data? And this really highlights that, yes, the Site Explorer viewer really is the complementary feature for that. Again, it has much higher compute power. It can do those requests, and these tools really should be used, yeah, in tandem. All right, we have another question in the Q&A. The first comment is, I'm really excited about the spatial data. Are there any future plans to extend the gene panel available for the cross-species data?
Yeah, so the 300 gene panels that we use for each species for the basal ganglia data collection, those will only we will only have those 300 genes because those are due to technical limitations of the current spatial transcriptomics platforms that were available at the time that we collected the data. So there won't be any more than those 300 genes for those data sets. It's not like we're holding anything back. Those are the ones we have. But, as these platforms are actively developing and, most of the platforms are pushing for larger and larger gene panels, that's clearly something that lots of folks are asking for. We'll definitely be taking advantage of those larger gene panels as we push to the whole brain as much as is technically feasible and available based on the platforms that we're using.
And then related to that, I know that for the whole mouse brain data, right, that initially was a 500 gene panel for the experiments done at the Allen Institute, and then eventually the single cell gene expression data was imputed onto that data set. Is that potentially a possibility for the basal ganglia data?
Yeah, that would be a possibility to do for the basal ganglia data as well. Although, kind of as we alluded to in our little tips, those analysis methods are also still very much in development, and so that's something that we want to think a little carefully about, because we certainly wouldn't want to misimpute a gene and make you think it should be somewhere that it's not. But yeah, that's definitely on the table to do it in the future, but just not right this moment.
Yeah, I think that's an important thing to emphasize, that everything is not set in stone. Again, with new developments in the new technologies, improvements in gene panels and analysis, things are always slightly improving. We have a couple minutes left. Let's see if we have any more questions in the chat. Another question that we have is, how do you think we can begin to connect in vitro functional properties with in vivo function of these neuronal types?
I can take that one. I think the goal ultimately is to understand what these neurons are doing in vivo, and right now what we have are sort of in vitro phenotypes, intrinsic properties, and things like that. But I think the way I would approach it is, I feel like optotagging is one of these gold standards. So there's a whole viral tools package that is also part of this effort. So I think that would allow people to target individual cell types and then record from them in vivo and start to ask, for instance, what would D1, D2 hybrid neurons be doing in vivo? Definitely harder in primate.
Yeah, and on that landing page that I showed, I didn't explicitly mention this, but the Genetic Tools Atlas is also linked on there as well, where you can see specific the viral tools that have been used to target cell types, specifically in basal ganglia. So that is also something that we did not highlight during that webinar, but these are tools that are actively being created and are also available for others to use in their labs as well. All right, so I believe that's we have one more question that we can do. Okay. Given the intrinsic electrophysiological features vary continuously across the dorsal ventral axis within the same transcriptomic type, how should we interpret differences in extracellular spike waves in vivo?
That's a very interesting question. I think what you're seeing is sort of these lower firing rates overall, wider spikes in ventral striatum. So yeah, that would be the difference I would expect extrapolating from these changes. I mean, they aren't huge differences, but that's what I would expect.
Okay, and that is it for our last question. I'm going to launch a very brief just feedback survey. It should appear on our attendee screens. This is just really helpful for us to plan webinars in the future. It's just a couple questions, so that really help us out for anyone in the audience to fill out those answers. And thank you everyone else for joining us today. This has been recorded. It will be on our YouTube channel, hopefully later this afternoon. And then you can have access to all those links that were presented during the talks as well. And if you're unable to fill out that feedback survey here in Zoom, it'll also be available as a link that'll be sent out in a follow-up email. All right, I'll just wait one more minute I see people are answering the surveys. Thank you very much. That's very helpful for us. And thank you again to all of our presenters today. We had a number of presenters from the Allen Institute and also outside of the Allen Institute. So thank you everyone for coordinating together to make this talk work. It can be difficult getting lots of people together for a big expansive talk like this, but I think it went really well. And just thank you everyone for being here today.